Have you ever been swimming or boating and noticed green plants at the bottom of a lake? Most people don't think too much about them, but these plants provide habitat for many tiny organisms called zooplankton. But what if that habitat was removed? Hi, my name is Purva Puri, and this summer I worked in the CLEAR lab under the supervision of Dr. Andrea Kirkwood. My research focuses on investigating zooplankton community responses to aquatic vegetation removal in Lakes Scugog and Canal Lake, which will form the foundation for my thesis. When we think about a lake ecosystem, we often think about fish, plants, or algae that we can see. However, some of the most important organisms are the ones that are too small to notice. Zooplankton are tiny aquatic animals that drift through the water and form an important connection within freshwater food webs. They feed on algae and organisms, and in turn, they provide a major food source for fish and other aquatic organisms. Because of this, changes in zooplankton communities can tell us a lot about what's happening within an ecosystem. My research is a part of the Trent Severn Aquatic Plant Experimental Removal, or TAPER project, which investigates how different aquatic plant removal methods influence freshwater ecosystems. The study takes place in Lake Scugog and Canal Lake and follows a before-after control impact, or BACI experimental design. This approach allows researchers to compare ecosystem conditions before and after vegetation removal, so things like pH, dissolved oxygen, water clarity, while also comparing treated sites with untreated sites. As part of this, my role was to process and analyze zooplankton samples collected from these lakes. I prepared samples using a Sedgwick rafter counting cell, which allowed me to concentrate and examine the organisms under a microscope, specifically a EVOS microscope. I then identified different zooplankton communities, including cladocerans, copepods, and rotifers. As I worked through each sample, I recorded my observations and organized the data in Excel, keeping track of factors such as sampling site, treatment, organism groups, measurements, and abundance. I also measured organism body sizes and used these measurements to estimate biomass through established length, weight, and biovolume relationships. You may be wondering why consider biomass. Biomass is important because simply knowing how many organisms are present does not tell us the full story. A lake with many small organisms may have a different ecological impact than a lake with many larger organisms. Biomass allows us to better understand the amount of living material present and how much energy may be available to move throughout the food web. By investigating how zooplankton communities respond to aquatic vegetation removal, my research aims to reveal the hidden connections within freshwater ecosystems. Understanding these responses can help inform better management strategies and ensure long-term health of our lakes.